I think we need to keep the meeting rolling along. It's now 11 o'clock. And I'd just like to introduce the next session. As we know, Ita is extremely well known for her groundbreaking work in B-cell and T-cell immunology. But her third great interest throughout her life was the macrophage. And we are very fortunate today to have three true leaders in the macrophage field, both in macrophage biology, differentiation, and function, to speak to us. So I'd like to welcome Emil Unanue as our first speaker.
Well, it's a joy to be here with all of you. And let me mention a few statements on Ita. So I was here, one of her first postdocs, I believe, in 66 and 68. I had come to actually work with John, who I had met when I was at the Scripps Research Institute, and he had come to visit. He was editing Advances in Immunology with Frank Dixon. And then I applied to him, and when I came to his office, he said, well, I think there's something important to work on macrophage. Anita is handling that. Why don't you go and work with her? Which I did for 2 years, and it was intense, I can say, and rather provocative, because we had made these simple observations. We didn't know how to actually handle macrophage, but then we purified them very well. I did have some experience with iodinated proteins, which I had developed in La Jolla, where I was. And these were rather simple, nowadays, crude experiments. We put the proteins on these macrophages, transfer them with lymphocytes, and long and behold, we got very, very nice and strong immune responses that were high and were reproducible. Well, we got very excited. I presented that for the first time at a BSI meeting. And then we went on and tried to correlate what was happening with the protein, culturing the macrophage, and it became apparent that most of the protein was being lost, but the immunogenicity persisted. And this was actually this paper in JXMed where this is an important paper. We actually followed the catabolism pretty faithfully, and we hadn't lost at all. So there was a molecule, an entity that persisted for long periods of time, and while the rest of the material was being catabolized. And Tony Allison then joined us, and we did some work with adjuvants like that. And you can see our sketch at this time. There was no MAC here, coming in, out, coming back out. And this was about the time of the Cambrian explosion of cellular immunology, you can call it that way. Hugh McDevitt had described here at Mill Hill the high and low responders to protein antigens that were associated with MAC. The first time that there was a physiological role for MAC described by him. This is at the NAS when Ita was inducted to the Academy. And at that time, there were the first reports coming out of T and B cells, and here we were with these strange observation that a classical phagocytic cell somehow did not degrade everything to amino acids, but actually had a protective entity. So it was a great 2 years. That sort of funneled my career, and that's what I did for all my time. And this was a long, long friendship. I have a huge correspondence with Ita. She was not as avid a writer as John was, but she had these little telegrams that she sent by email constantly, telling me, well, she's going swimming here or swimming there, or you're working too hard, please stop working hard. And that was one of her favorites to me. So it was enjoyable friendship with her. Or you're traveling too much, kind of thing. So this is what I'm going to be talking about. I'm not going to be talking about infectious disease. I've been talking about my interest in macrophage and in the context of antigen presentation. And I'll tell you two things. This one here, that islets of Langerhans contain a set of resident macrophages that live there, that are vital in maintaining a healthy functional islet. And then I'll take this into the context of what we believe is happening in the first early stages of the autoimmune diabetes of the NOD mouse. So this is not a section of islets of Langerhans. That's a whole culture of islets. So that's in 3 dimensions. You isolate the islets and look inside on what's happening. We've selected for this study a big islet in the mouse. And this is a non-diabetic mouse. And the point that I want to bring you out is here are the vessels. It's highly vascularized, as you would expect. And attached to this vessel is this network of typical macrophages. These are not dendritic cells. There are 2 things to note. Not only that they're firmly attached to the blood vessels, but they're also throwing dendrites inside the blood vessels. You can see some here, you can see some there, you can see some there, and so forth. And I'll show you a more precise one as we go about. So this is a normal population that inherits. We've examined islets of humans, islets of other strains, and they're always this set of cells. These are the cells of the endocrine organs that actually Simon Gordon described at 480 positive years ago. And if you take some of the features here, which I cannot explain, they're very long-lived. They're not turning over with monocytes. Actually, they replicate very, very slow. They're definitive hematopoiesis. In other words, these are not yolk sac-derived macrophages. Very interestingly, the yolk sac-derived macrophages of the pancreas that Frederick Geisman described are the ones in the stroma of the pancreas. So in the same organ, you have two distinct type of hematopoiesis going on. And the ones here in the islets actually are very from definitive hematopoiesis. These are the markers. They are not dendritic cells, ZBTB46 negative. They do depend on CSF1. So if you take the mouse that has the natural mutation of CSF1, these islets are collapsed. They're hypoinsulinemic. What is this homeostatic function? I don't know yet exactly how to explain it, but it's an important one, obviously. And these are the functions that they have that are relevant in the context of when you bring this into the NOD diabetes program, not only the attachment to the vessels and the intravessel dendrites, which we believe allows T cells to go in, but they capture secretory granules, and they're superb antigen-presenting cells to the products of the secretory granule. And that's what I will be developing with you. But this is a point that I was bringing. You can see them by confocal, these long extensions in here, that's in fast looking at you. So this is very, very consistent of them. And that this exposed, that area where here's a vessel in blue with pecan, and here is the macrophage in 11C positive, and you inject a bead, a 0.5 micron bead, which you actually coat with anti-IAG7, and they get stuck right here at that interface. So this interface, somehow, it's exposed to the circulation. Why is that physiologically? Why should that be an important role for this macrophage? I don't know. I think it must have some kind of a protective role at some point, but I don't know exactly how to define it at the moment, except it's very, very striking, that interface, how it is exposed. So let me go then back into the NOD mouse and what we've been doing in it. So what we've been looking at mostly is at the very early start of diabetogenesis. The NOD in our colony starts diabetics around the 16, 20, 24 weeks of age. But if you look at the islets and isolate islets around the 3rd to 4th week of life, you see your first flush of CD4 T cells coming into islets. And these are shown very nicely in this picture. This is an entire islet. Again, the vessels are in blue. And you see the CD4 T cells in red, penetrating now the parenchyma. You don't see the beta cells because we haven't stayed for insulin. And they are in contact, right, with these macrophages in the islets. You can see them here. You can see this area of contact right here. And they're all over the place. These ones, that one, that one. There's 4 of them here in this area here. So this is a very intimate contact of the first flush of CD4 that comes into islets. These are anti-insulin reactive T cells. So if you have a situation where you have no insulin expression because you have made mutated insulin, so the immunogenic component is not there, these T cells do not enter the islet. So we're trying to sell the issue that this is the first event that programs the entire development of the process, and that's our interest in trying to understand as much as possible this early phase. At this first time, too, when these T cells come in, there's a flush of interferon type 1, an interferon 1 signature. You can see it by isolating the islet beta cells. There's a constitutive one, but already at 4 weeks, and from there on, you have a flush of interferon beta in this slide, also alpha, and interferon gamma comes a little bit later. So the point in here then has been to try and explain this insulin reactivity in face of what we know insulin is presented in the thymus. Because what we know, here's insulin, which is made as a prohormone. It reaches the secretory granules as a hexamer and is further processed by a number of very well-defined proteolytic steps until the mature molecule of 51 amino acids. And what we know from the work of George Eisenbarth, and he really was a champion on bringing insulin as a major molecule that we had to pay attention to. His group first described in '94 that when you isolate T cells from the islets of Langerhans or the pancreatic nodes, you found a lot of anti-insulin T cells that transfer disease. And most of them were reacting to this segment here of the B chain of insulin, which we've diagrammed here in that little red bar. What I will show you as you actually get from lymph nodes and particularly pancreatic nodes and make a large bank of T cell hybridomas and examine them, the majority of them are against this particular segment in here, while the minority are against this particular segment in here. But the conundrum is, we know that insulin is present in thymic epithelial cell. This is a picture for Bruno Kiewski, where he actually has done beautiful studies, molecular and so forth. When he isolates MTEC, they are producing insulin. And this is a picture of him actually detecting it inside the MTECs. We know that if you have mutations in air, you have the monogenic autoimmune diseases in which you get actually a lot of endocrine autoimmunity, including diabetes, saying again that the insulin in the thymus was functional. And in the human, while the odds score for the class 2 HLA-DQ8 is around 20, the second more susceptible gene is insulin. It's about odds score of 2. And that is that there's an allele which determines the level of presentation of insulin in the thymus. And that allele in which there's lower level of insulin presentation in the thymus is the one that you have more susceptibility to diabetes. So this was the conundrum. How then do we explain that indeed you have higher reactivity to insulin despite the fact that you know you have control in the thymus? And that's when we started looking at the specificity of the T cells that actually were found in the islets. And in the next 2 slides, I'm going to summarize what is a very large number of experiments with synthetic peptides of different sizes and mutations, which I am going to skip completely. I want to give you the main message of these. So what you find then, the majority of the T cells that you're finding have this particular feature. This is an antigen presentation cell assay. You've isolated them, bring them ex vivo with APCs, and now you do your reaction. You give them the long peptide and they react nicely, but you give them insulin and there's nothing. Well, this is the typical, the kosher antigen presented CD4 T cell. They see insulin and they see the peptide. Okay? These are less abundant. These are the ones deleted in the thymus. These are the frequent ones. This is not an artifact of having a synthetic peptide. The same reactivity is to denatured insulin. In fact, we made 11 clones of this and they all transfer diabetes. So the cells that we were finding spontaneously, okay, had this particular feature in here of poor reactivity or none to insulin and this. As we went further in trying to understand what this was all about, it became evident that these cells in here, okay, reacted with this segment of the B chain with glycine at P9, which is a very weak binding segment. While these cells in here, the good ones, had a at P9, and this is a very good binding segment. How we actually told that is by we constructed APC lines in which in the beta chain you had either 1220 with glycine or 1321 with glutamic acid. And then took these APC lines and checked all of these. These ones over here see 1220, doesn't see the other way around. Okay. So there's a feature in here, one amino acid register shift decides what population of T cells have evaded negative selection. And these ones, which happen to be very weak binders, are the ones that have escaped. And they're not recognizing insulin as we know insulin being processed. So these are some of the features of it in summary. They only recognize exogenous peptide or denatured INS. They evade thymus negative selection, and they enter islets spontaneously to interact with the islet macrophages, which are heavily loaded with peptide MAC complex. We just did a TCR transgenic mice of one of these. It was just published in the Journal of Experimental Medicine. And there are a few points to bring out about these cells. And it's interesting to compare to a second TCR transgenic that has been made with Mark Anderson. We're doing this collaboratively in San Francisco, which is a type A. These ones here, which are the ones that are against that particular peptide, enter the islets naturally. By 3 weeks, they are there. You don't need pancreatic nodes to enter. Without pancreatic nodes, they enter perfectly all right. Okay? And they are diabetic. As such, they produce a lot of inflammation, but they have normally a lot of endogenous TCR alpha and Treg component. Okay? If you put them back into a rack, rapidly they are diabetogenic. Okay? Or if you have a rack that has no pancreatic lymph node, they're also diabetogenic. So these are bioactive cells that are leaving the thymus. And if you look at thymus differentiation with them, you can see that there's absolutely no negative selection taking place in there. And the last thing in here is that they're actually helper cells, because they induce actually the formation of anti-insulin antibody, and they're anti-native antibodies. These mice have a lot of anti-native insulin antibodies that you can pick up in serum. And moreover, and what I find it fascinating, there is deposit of antibodies within the islet of Langerhans. This is what it shows in here. You can see them in the vessel wall as little beads. I would bet that these are complexes with insulin. So in other words, these T cells can actually break through. They are not eliminated in the thymus. They enter the islet spontaneously, and they seem to be driving the response. You don't have these early T cells, and the diabetogenesis stops cold. So this is what we're trying to do right now. We're trying to explain how the presentation of INS peptide differs from the presentation of INS protein, an issue that I have been very interested for some years, because we had found this kind of phenomena with actually protein antigens, typical protein antigens, these kinds of features of one set of T cells seeing peptide and not protein, except in here, we didn't have to select. This was spontaneous, where these kinds of cells were coming up. And the second point is, if you believe that it's this insulin peptide that's driving it, where is that insulin peptide being generated? And of course, you have to go to the islets of Langerhans. And that's what I'm going to summarize the work that has been done along this line. So this has to do then with the issue of the actual processing of the insulin protein. So take a look at these two peptides in here. This is the weak binding peptide that has glycine. Very critically, in the IAG7 molecule or DQ8, acidic amino acid at P9 is actually the critical residue. Since you don't have aspartic 57 in the beta chain, you actually form the salt bridge with the unpaired arginine on the alpha chain by having an acidic amino acid. So if you compare now the binding and the AK off, this is I-125 peptide bound on G7 and letting it go, you can see that 1220 is kicked off very, very fast. You add to the reaction DM, and practically you have no binding whatsoever. DN is the auxiliary molecule that is in the deep compartment. While this peptide here is a stable peptide. Okay, it has a very prolonged stay as a bound peptide. And you can also do this by SDS. You put SDS of the long peptide radioactive, it's here. When you don't boil, it's stable. Boil it and it kicks off. But this peptide in here in itself falls off right away. All of these tests is telling you that this is a good stable structure, this is a weak structure. So what happens then is that insulin is taken to a deep intracellular compartment where all proteins are taken. They all have to be denatured and opened up. You have 2 disulfides in insulin. By the way, this has been done in a number of things, throwing materials in different compartments. This is just a summary. And in the deep compartment, you have this editing molecule H2DM. This molecule here is going to kick off this peptide. So in the deep compartment, you're going to see alive only this particular peptide. That is the only one that survives the processing in the deep compartment. But when you bring in exogenous peptide, you never actually, or denatured protein, they never reach the intracellular vesicle. They're in endocytic compartments that are recycling. And there, you can give rise to whatever epitopes you have around that area of the molecule. In other words, you in the deep compartment constrain the repertoire. In the recycling compartment, that epitope is not constrained. It's much more open. And you are selecting certain peptides which have very interesting biology, like this one in here, because actually the T cell to that peptide are not actually perched in the thymus. So this is what we believe is happening insofar as the handling, and why these T cells, when you give this peptide, okay, you react with it, but when you give insulin, you don't see it because it's eliminated here on the deep compartment. And again, a number of manipulations of cell biology is pointing to that which I am not actually detailing to you. But then this is the last part that I want to mention about, which is our great interest right now. If you're saying that these T cells are reacting with denatured insulin and peptide, where are these coming from? And the point is that if you harvest these islet macrophages and purify them, you have to isolate 20 mice. My budget for NOD mice is rather big. I need help. And you have to isolate about a dozen or 20 mice, have enough islets to break, flow sort, and isolate your cells. And indeed, they are extremely effective antigen-presenting cells. Okay. They have a mean of about 10 granules captured per cell, and each granule has about 10 to the 6 molecules of insulin. Make your math. That's why they're extremely loaded with PMAC inside the islets, nowhere else. But when you look at the handling of insulin, this is the typical procedure that has been very, very well worked out by excellent cell biologists. So this is the regulated pathway that when you have high glucose, there's changes in calcium, the granule is pumped out, so you have a regulated secretion in here. But for a long time, beta cell biologists have been talking about this pathway here, where they say, indeed, out of the regulated secretory pathway, there is a bypass pathway in here which is under control depending on the amount of insulin that you're making. And particularly because this is one of the highest secretory molecules in biology, you're going very easily into endoplasmic reticulum stress. And by doing that, you are shutting off the actual regulated pathway and going to a bypass pathway. So this is in their books. They actually use a special name for these vesicles. They call them crinophagic vesicles. So these are it. As soon as we were looking into that, our thinking was, well, maybe these are the ones that contain the antigen that we're talking about. These ones were the ones that may have denatured insulin or peptides from insulin. So what we made in here is a monoclonal antibody to the peptide. That same peptide that the T cell recognizes, we made a MOAB to it. It doesn't see insulin. It only sees peptide. And with that, we are then going inside the cell and trying to fish out whether we can find these peptides. And they're here. This is a whole islet, only stained with this monoclonal, and you can see that it's full of granules with this particular peptide. This, we believe, is the same pathway that we're talking about. Okay. We've isolated chemically, and as you would expect, if you isolate secretory molecules, the majority of them have INS and C peptide, but there's a bunch of peptides along the B chain and also the alpha chain, which we believe are these immunogens that are actually firing the T cell response. So you can document this in a number of ways. This is, for example, one of these cells next to two beta cells. Look at the extraordinary amount of membrane activity that's going on in here. There's an interaction in here which I don't know what is going on, but there is something here. This is not just randomness. And you see then the entrance of some actual granules inside these cells, which you can pick up with your antibody as you have it. So these two cells enter contact. That's what we're studying. We have an insulinoma where we can get the granule labeled with GFP, and as you put the two cells together, you can see the transfer of these vesicles into them, and the dynamics of this is the issue that we are now very much examining. So this is what I've told you, that we have evidence that beta cells contain abnormal catabolites of insulin as a result of processing of the prohormone, that these are released and passed to resident APCs to generate peptide MAC complexes that select for unique T cells that have bypassed thymic control. And we believe that they may be responsible for initiating the autoreactivity. Does this apply to other endocrine autoimmunities? I am sure of that, because there's a common denominator in all of the hormone tissues. A prohormone made on a secretory granule that has a lot of proteolytic enzymes, and always a network of APC surrounding the hormone site. So this is our view from St. Louis then, that you have an entrance of a first stage of T cells to the islets, these are the ones we believe I've described to you. Immediately you have an amplification of the process, the pancreatic node amplifies, now you have epitope spreading, and now it's very hard to control the whole disease process. So this is the group that has done most of the work that I'm very proud of. Boris Calderon did a lot of the cytology, and Javier did most of the genetics of the studies, as well as Stephen and Jim and the other groups in here. So since I'm a bit ignorant in that sense, I make all of this fit into actually this kind of a pattern. If you are limited in what you overall know. We should all read this book, by the way. Makes us humble after reading it.
So we have time for some questions then, or maybe I can start. So your results would imply that a NOD mouse without lymph nodes should get disease. But I don't think that's the case, is that correct?
No, that's not the case.
So how do you square that with what you...
Because
Okay. So we're just discussing the fact that NOD mice require lymph nodes, and the implication of Emil's results would be that they may not. But
no, I have a slide on this, and I'm not projecting it. And you need the pancreatic node as an amplification step. There's no question about it. But if, on the other hand, you don't have these first T cells coming into the islet, that process stops absolutely cold. If you don't have the pancreatic node and you look at the islet, you do get the flush of these first T cells in. You get them in, you start amplifying, but you don't go beyond that. So there has to be cooperativity between this and the next step in the reaction, which is actually represented in whatever is going on in the pancreatic node. CD8 T cells are important then coming into the reaction.
Nick was first.
Do you think these processes from the presenting cells protruding into the lumen of the blood vessels might also happen in the brain? Because I always think it's amazing what an strong sense of smell myelin-specific T cells seem to have when they're homing to cause EAE in the brain.
Yeah, you know, that has been shown in the aorta, that has been shown in the intestine. In the intestine, you know, you can say, well, that's why they're there, to pick up some antigens from the lumen and bring them in. I can't see a beta cell doing this here because it'll harm that. That beta cell, you don't want to have an immunological reaction in the islet. On the other hand, either it's a paraphenomenon and forget about it, or it may be something more interesting that we have to find out what it is.
Emil, I've always been struck by the lack of complete penetrance of the disease in non-mice, which are totally genetically identical, and it's much more penetrant in females than males. Have you looked at any of the mice that survive their whole lifetime without penetrance to see at what stage in the process? Do they start the process and abort it at some point, or does it not start?
Yes, if you, if you look at your colony, and depends how good is your mouse house.
Yes.
Ours is very clean, it's 85–90%. I'm sure that it had been the Mill Hill, at the time that I was there, it would have been 20%, you know, because that animal house was. But the remaining of the mice, even though they're not diabetic, they do show inflammation. I mean, those islets have different degrees of inflammatory reactivity. They're not pristine islets, far from it. Okay.
Andreas.
As in these unusual catabolites, do they actually never reach the thymus and are not presented? Or is there another story of why the T cells escape negative selection?
I don't think they reach the blood. I think they're very locally produced in there. Okay, so, I mean, there's no doubt you are not tolerant to them. Forget about diabetes. You take a young NOD mice or a pre-diabetic male mice. You immunize with insulin like you're immunizing with any protein, and you do an ELISpot day 7. Zero reactivity whatsoever. Zero. You immunize with the 13 to 21 peptide, the component of insulin. Zero reactivity. You immunize with 1220. Boom, 200 ELISpot positive. Okay. Very clean. So you have no reactivity to that here. You only have it to that segment.
So 3 very quick questions and quick answers. Colin.
Is it possible that the stringency of peptide editing is different in the thymus and in the pancreas?
I'm sure of that. I don't think in the that's a very good point. I don't think in the pancreatic, in the thymic epithelial cell, you have the proteases that regulate the processing of insulin. You don't have them there. So whatever way you are handling and processing that protein is very different from the beta cell. We are trying to do that now, except it's not very easy to get enough MTechs to do this kind of thing, but we're going to try.
But the levels of DM, sorry, the levels of DM in the two places are the same or different?
I can't tell you that. Yeah.
Do you think the HLA association in humans with diabetes with the HLA-DQ2 and DQ8 is to do with the macrophages in the pancreas preferentially expressing that type of class 2 of ER? And very brief on top of that, why do you think so many inflammatory conditions of the pancreas that humans get don't trigger diabetes?
Well, let me get, of course that's key. I think the issue of the bypass pathway on beta cell is in every beta cell. Non-diabetogenic, we've taken human islets, now we've got 3 beautiful experiments on isolated clean, and these peptides are there. The second part of the equation is your cell that has the right HLA and the other genetic components that then drive that process after they've taken these peptides. I think those are the key things. And in my view, from all these things, I think the central cell is that intra-islet APC that actually takes those peptides and then starts the whole process.
HLA-DQ expression as macrophages.
Oh, they do express it very nicely. No doubt about it.
So my question then follows actually perfectly, thanks for the inflammatory mention, because, okay, so these peptides are there, you have genetics with a predisposition, but still you know even insult to the body by an infection that may all of a sudden reveal a lot more of those peptides in the wrong context that
Yes.
then brings about disease?
Absolutely. Absolutely. You can think on these. I can turn around the identical twin stories the other way around, by the way. The one that gets diabetes is the clean one. Okay? While the one that doesn't get diabetes is the infected one. Remember, these mice in autobiotic situation are not only diabetic, but the penetrance is higher. So, you can play the game both sides. But nevertheless, there has to be an incident in here, as you correctly say, that turns on the system one way or another.
And maybe a little bit of dirt. I think that Anne Cook actually has postulated that certain infections will actually downregulate diabetes. Maybe that the dirt and
Exactly.
the infection may put it the other way.
I'm afraid we need to move on now. Simon? Thanks.
Grab the slide, please. Okay. Right, well, I'm also privileged to be here and could have, okay, I'll start the next slide. So this is the time I really became friendly with Ita. We were both at a meeting organized by Manfred Karnofsky to celebrate the centenary of Mechnikov's description of phagocytosis at Messina. And both Ita and Philip Darcy Hart and I went off on a tour of Sicily after this meeting, where we stayed at Siracusa and had a great time. And you notice from the paper over here that she presented that David Sachs apparently was there as well, and he tells me he was. Now, the kind of work that we've done is mostly on the non-opsonic receptors of macrophages. Apart from the antibody and complement receptors, which of course are very important, they do express a whole range of different molecules, and I've picked on a few here. First of all, there are the dreaded toll-like receptors, which we try to avoid in a toll-free zone. Then we have regulatory receptors that either enhance myeloid cell responses or diminish it. The ones that enhance it are like the TREM1 family, but we also worked on the F480 molecule is part of a small family of myeloid adhesion GPCRs. The F480 antigen was a prototypical molecule. So that will downregulate some responses or upregulate depending on the circumstances. And then we have a negative system to control macrophage activation, the CD200 receptor pair. These are Ig superfamily molecules. But then we also have a group of scavenger receptors like SRA1 or MARCO, two different gene products, and these are collagenous transmembrane molecules. And then we have a different kind of scavenger receptor, so-called class B, the CD36. It's hard to call these a family if the molecules look so different, but they do have something in common. They like apoptotic cells, they like polyanions, they are very promiscuous in their activity, and I'll say a little bit more about that. And then we have a group of lectins, and I've picked two out of many. There's one called dectin-1, which has an ITAM motif, and that is important in fungal recognition, the beta-glucan receptor. And then we have a multi-lectin called the MANNOSE receptor, which is also more subtle. It has 8 of these carbohydrate recognition domains, and it has an N-terminal cysteine-rich domain that's interesting, and I'll say a little bit more about that because that was the clue to us to describe a form of macrophage activation that is alternative to the activation pathway induced by interferon gamma. So first I'll say a little bit about dectin-1. This was discovered or described by Gordon Brown in my lab. And what we have is an expression cloning which showed that this was the receptor for zymazan, it's unopsonized, and is now known to be both in mouse and in human. Here's the ITAM motif. And then it's found on macrophages, dendritic cells, and neutrophils. It's not only responsible for zymazan recognition and uptake, but pretty much for many fungi, including C. albicans and pneumocystis. Now, the secretion of TNF, the cytokine response following, say, zymazan stimulation, has shown us that there's a collaboration between a TLR and the lectin. And then there's another kind of a sugar-independent interaction which we have not been able to discover. So this was the first example we had of collaboration between a toll-like receptor and a lectin-like receptor, like lectin. And of course, the toll-like receptor uses the MYD88 pathway. The lectin works through SIC, and then there's also various downstream molecules that have been implicated, and that will influence cytokine and chemokine production. Now, this is an important theme, and you can see that you need both the lectin and the TLR2 signal to actually get an effective, in this case, TNF response. And I want to, this is a slide given to me by Gordon Brown, who's worked with Brazilian collaborators in a quite uncommon, a chronic fungal infection. It's a chromoblastomycosis. And you can see that in this case, the lectin needs to collaborate better with the TLR. And what they found that the treatment for this chronic indolent ulcer is actually to use imicramide to stimulate TLR function. And that gives you quite dramatic improvement in the skin. In any case, it's not our work. So I want to turn back to the scavenger receptor now, the SRA. And it was implicated originally by Brown and Goldstein and Monty Krieger and Tat Kadama as an important receptor for modified low-density lipoprotein. But we, by chance, made an antibody against the scavenger receptor when we were looking for adhesion function. And then we thought that perhaps the receptor does not exist for recognizing modified lipoprotein and playing a role in atherogenesis where it does, but it must be contributing to innate immunity. And so we screened many bacteria, and we found that the Neisseria were exquisitely specific and selective ligands for this. And this is shown here, where we have wild type and knockout macrophages. These are bone marrow culture-derived macrophages. This is the B group Neisseria meningitidis. And you see very striking binding. This is in the absence of antibody and complement, missing in the knockout. And this is specific because the specific antibody we had blocked it, as did non-specific polyanions, like polyinosinic acid, but not unrelated antibodies. Now, all the strains we looked at, whether they were pathogenic or lab strains and so on, were actually ligands. And this is a very immunomodulatory, a very potent immunomodulator. The vesicles derived from the Neisseria as well. Potent inducers of pro-inflammatory cytokines, but the recognition and uptake of the Neisseria was uncoupled from the ability to produce TNF. And the ligand was not lipid A, which is what we had expected, but they were actually, this is needed for the cytokine release, but the ligand we were recognizing, that the scavenger receptor was recognized, were actually bacterial proteins. And I think this theme remains to be explored in many other situations. So the scavenger receptor, I've summarized, is an important non-opsonic receptor. It's actually true also for Neisseria gonococcus. There are 3 of these proteins in the bacterial wall, and these were not known for any particular function, but they're very highly conserved. They're found in all these strains, and including in the gonococcus. And we've made knockouts of these bacterial strains. When I say we, this is Richard, of course, in Oxford. And then there was no obvious phenotype in vitro, but we found that in vivo it did make a difference. And so it's possible that these are not so-called pathogen recognition receptors, but they really see kind of molecules that are found in commensals and even in other membrane molecules. So it is protective to have the SRA in terms of the septicemic phase. You can't model the Neisseria infection as we see it in humans in mice, but you can model the bacteremic phase, septicemic phase, and you need to inject a source of iron like transferrin, and then you can see that you can set up a mouse model, and the presence of the SRA is able to protect the mouse to some extent. So I've mentioned this because we still don't understand why only a sub-fraction of humans actually develop the meningitis following invasion of the nasopharynx. Now, we used the meningococcal septicemic model again to explore the function of CD200 and the CD200 knockout mice. So you can see if you do the infection, you find that the mice seem to do very similarly up to about 40 hours, and then they splay, and that the knockout mice, which are supposed to be negative regulators of an inflammatory response, actually then, in the absence of that negative regulator, will die much more readily, actually, than the wild type. And the bacterial load is no different. And if you look at another cytokine, in this case, or rather health status, you see again that the CD200 plays an important role in down-regulating the effect of the organism. Now, we've looked at this in some more detail, and we see that both TLR and NOD-like receptors, as well as the inflammasome activation pathway, all of these are controlled, apparently, by the surface interaction by CD200 and CD200 receptors. I want to turn now to a different organism, which again macrophage people tend to neglect. We all think that Streptococcus pyogenes depends mainly on neutrophils and complement for resistance, certainly in acute infection. And the most striking molecule there is the M protein, which really taught us, as understood all of immunology, if you like. The molecule itself has hypervariable regions and other conserved regions, and many of the immunologic ligands you'll see in this list interacting with different domains within this molecule. And we found there are more than 120 different M serotypes, and I believe that it's still not known how this protein is actually, how this is genetically controlled. This is a very important virulence factor for the bacterium, because antibodies will, directed against this, will actually protect the host. So Thomas Arashuk visited my lab. He was interested in the organism, and we started asking whether the scavenger receptor could recognize this particular organism. Well, it turned out, and I'll just summarize the main result, that if you look at the key ligand, the classic gold standard ligand, the acetylated LDL, you get striking binding, which is absent in the case of the knockout. When we look at the organism, and this is an M5-bearing organism called Manfredo, then you find that there's no ability to discriminate. But the organism seems to use this protein to mask the expression, because if you delete this bacterial protein, the M5, now the scavenger receptor binding is revealed. So this is an example of bacterial evasion of recognition through the scavenger receptor. Again, I think that many other extracellular pyogenic organisms do interact with macrophages, and we shouldn't forget that. I want to turn now to the MANNOSE receptor. That is how we discovered or described a process of alternative macrophage activation. And this is important in limiting excessive inflammatory reaction, and especially in host defense against parasitic infection, classic Th2 diseases. So the properties of these alternatively activated macrophages, you get upregulation of the MANNOSE receptor, whereas with gamma interferon, the classic activation, you get downregulation. It really splays them apart. You also get upregulation of the MHC class II antigen, and suggesting to us that this was an activation phenotype and not a deactivating one. Instead of INOS, the nitric oxide producing system, as you find in classical activation, you find upregulation of arginase 1. And you get marker proteins such as chitinase-like proteins, YM1 and 2, and a resistant-like protein, FIS1. These are markers in the mouse that are very useful in distinguishing. Now, what I haven't shown you here is that the key cytokine that would mediate this is IL-4, and its its analog, if you like, IL-13. Now, as a result of treatment of macrophages with these cytokines, you get a release of selective chemokines, for example, CCL17 and 22, and a phenomenon I'll come back to called macrophage fusion. And we tried very hard. Several student half-lives were used to try and search for specific surface markers for these alternatively activated macrophages, but we were unsuccessful because they're all quantitative differences. And the important message is that we are dealing with species differences. Human and mouse are not the same. And these markers do not necessarily apply. And this was the MANNOSE receptor structure I alluded to earlier. So this is the C-type lectin activity. 8 of these carbohydrate recognition domains, you see mannose, fucose, or glucnac. And these are found in a whole range of different organisms, both mycobacteria, for example, HIV. We've described this on dengue virus. It recognizes the dengue virus glycoprotein, Candida and pneumocystis. Now, this is exogenous or foreign, but you also see many host molecules that happen to have the right terminal sugars. All the lysosomal hydrolases lose the phosphate after secretion, and then they express the terminal mannose. Neutrophil degranulation product, myeloperoxidase, will express this, and so will TPA, the immunogen activator made by the endothelium, and even hormones will also express these. So here the point is that these are not foreign molecules, they happen to be in the wrong compartment, and therefore, for example, in the plasma or extracellular fluid, and they have to be cleared from the circulation, and that's the key function of macrophages, a homeostatic clearance function. So here we have dual recognition, if you like, modified self, inappropriate self, and foreign. But if you look at the N-terminus, here's a highly conserved cysteine-rich domain, and it turned out that this was a second lectin for sulfated sugars of this structure. This does not interact with microbes, but again, there's self-recognition, hormones again, for example, the anterior pituitary hormone, lutropin. So again, an endocytic clearance function from this. And I won't have time to discuss the function of this molecule, but it's much more than just an adhesion endocytic or possibly phagocytic receptor. So that had been the clue, and it's true in humans as well as in mouse, that it'll indicate that the macrophage that expresses high levels of this probably or could be under the influence of the Th2 cytokines I've mentioned. But we set out to produce more markers for the human because there are a lot of pathologies, and I won't have time to discuss this, but there was a lot of loose thinking about what alternative activation might be. So we took all the conventional culture models in human derived from blood, which is convenient, or in the mouse from bone marrow or peritoneum, with or without an inflammatory stimuli. And then we grew them on different surfaces. So this encompasses pretty much all the conventional models. And then we asked by microarray and proteomic analysis, what could we find a molecule or molecules that were conserved throughout these different conditions? And it turned out that there was one molecule that was highly conserved for both human and mouse alternatively activated macrophages, both been looking at the message and the protein. And this turned out to be transglutaminase 2, which is a transaminase which cross-links many substrates. It's like factor 13. And it's found not so much on the plasma membrane, but also in the cytoplasm and the extracellular matrix. And it has been implicated in autoantibody diseases such as celiac disease, gluten sensitivity, and in a nephrotic syndrome. So we wanted to validate this. So we looked at a classic Th2 type model in human now and in mouse for comparison. And this is an asthma model, an asthma patient. So here's the macrophage marker CD68 co-expressed with the TGM2. Here's in the mouse, the CD68 and TGM2. And you find pretty good co-localization. If you look at the monocytes, you find that there's unusually expression of this antigen or this marker already in the actual monocyte population. And there too, there's a negative population and a positive population. As if the body already knows what's going on in the peripheral tissues. It's an intriguing possibility. And then we validated that these are truly asthmatic subjects, eosinophil counts, exhaled nitric oxide, and so on. And the other beauty of the system is that the expression can be both following the antigen and the enzymatic activity. So I'm trying to get people to apply this to their own models of disease. But we've looked at the schistosome egg granuloma model, and you know that this is a very strong Th2 response at a peak. Here's the fibrotic reaction to the egg, which is in the middle of this granuloma. And in this case, we find that this is autofluorescence. You should ignore that. There are lots of macrophages seen around this. This is in a mouse model now. But yet, the TGM2 is only on the periphery of this granuloma, which is where the fibrotic reaction would be most prominent. So that's, I guess, guilt by association. We think is relevant to that. So now what we're trying to do is to get to grips with the idea of plasticity of macrophage gene expression. And again, we've done mostly microarray analysis, and we're looking at the effects of co-stimulating the macrophages, not just with the cytokine alone, like IL-4, but then challenging it with a pathogen afterwards. So now we're mixing things up more like real life, if you like, multiple stimuli rather than an individual stimulus. Now we've used different organisms, but in this case, Here we have Neisseria as a TLR stimulus. If you look at this closely, we start off with resting cells, and then with the Neisseria, you get a potent TLR-induced set of genes expressed. If you use AL4 by itself, you get a different set of genes. But if you pre-treat with AL4 and then follow by the pathogens, you find that you retain most of the molecules already induced by these in isolation, but you lose a few. So we call this chimerism. This is a chimeric signature, and you can, we're trying to reconstruct the effect of various multiple stimuli. I've only used 2, but we've gone up to 4, to see if we can find traces of the earlier encounter in terms of gene expression. And that explains why, or could explain, why these aren't simple buying two, it's not just one or other, it's not just M1 or M2 as people are saying. And we've compared this now with, here's Neisseria, we've also used Leishmania and H5N1 influenza to look for overlap and to see what is common to the organism and what's common to the macrophage. So now in the last few minutes, so I have 5-10 minutes, is the giant cell phenomenon. Now, when I was a student, I worked on Sendai-induced fusion of melanoma cells, in this case with macrophages. And of course, we are interested in the fusogenic properties of the virus, in this case, Sendai. But more recent, and this is something that happens in the body physiologically, for example, or pathologically in response to foreign bodies like hair fibers. It's physiological in that the osteoclast is a macrophage giant cell or monocyte-derived cell, which degrades living bone. And it's been known for many years that in granulomata, for example, tuberculosis, you find these beautiful Langerhans giant cells. So we soon found out, or as it was reported by others as well, that AL4 and AL13 will induce giant cell formation in culture. This is a single cell in vitro. And this was then important near the end of my lab career to go back to this original model and try and find out what are the mechanisms of fusion, and if possible, are there any functional consequences. So we set up a quantitative assay for fusion. We take peritoneal macrophages, we divide them and label them differentially, and we co-culture them with the AL4 present, and you find they go yellow when it's red and green fused cells. And you can quantify that, and you can see it microscopically, and you can see that in this case, the AL4 receptor is critical, because in its absence, you lose it compared with some background fusion. Now, we use this system to make monoclonal antibodies to find out if we could probe the surfaces of these fusogenic cells. So in this case, we were looking for antibodies that would either enhance the fusion, as in this case, or actually inhibit it. And one of these was actually tetraspanin CD9, which has been implicated in sperm-egg fusion. So there are properties in common between macrophages and other fusion systems in the body. But more interesting here is we had a whole group of independent isolations, and all of them were against one antigen, and that was the scavenger receptor B, scavenger receptor CD36. So we showed that you needed the CD36. If you had a knockout, you lose the fusion. This is shown here, and that the antibody was responsible for that. We also wanted to look at the purified cells so we've explored the CD36 phenomenon in some detail, and you need it on only one of the cell partners that's fusing. You don't need it on both. In other fusion models, you need a molecule to be present on both. So now we want to try and understand a bit more about why this happens, and what are the properties of the isolated giant cell, or the giant cell. So we purified some of these by a simple sieving procedure. Here are the giant cells. Here's the unfused cells. And we looked for the expression of markers like arginase, which is upregulated by AL4, but it's not differentially upregulated or altered in the multinucleated cell compared with the unfused AL4-treated cell. On the other hand, other molecules, other markers, such as the metalloproteinase 13, was actually selectively enhanced in this case. Now, we've done this more with and asked whether the LPS response, for example, following LPS treatment, whether that's changed, and actually it's reduced in these giant cells relative to the unfused cells. Less TNF message, less protein. Now we can go back to these granulomata, for example, in the schistosome model. You can laser microdissect them. Here's one that's been cut out. And we can replicate what we found with the metalloproteinase, that it was increased in the giant cell compared with the unfused cell, whereas arginase activity was not affected. So that was secretory function. What about endocytic or phagocytic function? An FC receptor mediated uptake, for example, is not affected. If anything, is reduced. Zymosan uptake is not affected. But we found, surprisingly, that these cells had selective enhancement, the giant cells, for CR3 function. Now, the normal macrophage will not, and the resident macrophage will not bind and ingest actually complement-coated red cells. Until you treat them with PMA, then you can induce it. But we found that in the giant cell, even if you hadn't treated with PMA, they were already able to eat these particles. So even in the absence of external activation stimuli. So again, we have properties that are enhanced, properties that are unaffected, and properties that are reduced. So these cells are fairly different in their actual function. Now, an obvious question is, you have these giant cells, do they take bigger particles? So we've done systematic screening, and here's an example, 20 micron beads eaten by the thioglycolate macrophages that have been fused, and you see they're much better at it than the unfused cells. So there is some preference, and there's some evidence that they utilize the size and the villi on the membrane more effectively than the unfused cells. So these are the people who've done the work I've described in passing. I don't have time to go into all the details, but I wanted to show you something else we've done, which is to look at prevention of HIV in South Africa, where I come from. With the help of Fran Borkwell and Mick Rolfe, we produced this cartoon book aimed at 12-year-olds called Staying Alive, Fighting HIV, AIDS. And a second edition, we revised it quite a bit, called The You, Me and HIV, hoping, if you like, that if you people understand what happens in HIV, that they will actually take the appropriate measures to prevent infection. I can discuss this. The book was published by John Inglis at the Cold Spring Harbor Laboratory Press. We've distributed about 100,000 copies in South Africa, and I still have a social worker in the KwaZulu-Natal area who's helping by having sessions with teachers and with health workers, and of course with adolescents. Thank you very much.
So Simon, in celiac disease, is the transglutaminase coming from alternatively activated macrophages?
I should have said that it's not cell-specific, and that initially we always wanted reagents like monoclonals that would only bind to macrophages and not to anything else. So transglutaminase 2 is an example of a molecule that is expressed on other cell types, fibroblasts, but is clearly upregulated and usefully so in the activated macrophage.
Other questions?
Transglutaminase 2 in the classical activated macrophage, or only in the...
I didn't show you the validation at the cellular molecular method, but of course we were looking for markers that are really valuable for this kind of activation, alternative activation. So it's not a useful marker in the classically activated. So if you treat with gamma interferon, you don't see that.
You don't see it.
I mean, okay.
Any other questions? Yeah, Rick.
Do you think transglutaminase is involved in wound healing? So one of his,
Well, everybody assumes that alternatively activated macrophages are involved in parasite infection, as you know well, but also in repair processes, and I mentioned the metalloproteinases and the hinting at the fibrosis and so on. There's actually almost no evidence that that these cells do play a role in repair process, and in fact we're doing an experiment with Judy Allen now and Paul Martin to test that idea, so well done.
Okay, thank you very much, Simon. Greg Bancroft.
So thank you. It's a real honour to speak at this symposium. And I think it's appropriate on a day like today to give acknowledgements right at the beginning. It's been my good fortune to study with and work for three very special people. On the left, my boss in Western Australia, Jeff Shellam, and of course, Ita, in the centre of my slide and really the centre of our day today and tomorrow. I was not always surfing on the beaches of Perth, Western Australia, but I did leave the beaches of Perth to join Ita's lab in 1980. And I have to say the weather is pretty rubbish in Mill Hill, but the science was fantastic. And Ita welcomed me to a group who wasn't actually working on viral infections, but it's important to remember that Ita also, at that time, was interested in parasites, and she actually had a group with David Sachs and Clem Groskinski and others working on African sleeping sickness, and I think that started me and my interest in tropical diseases. And then Ita guided me on to Emily Nanaway's lab, and eventually I ended up here at the London School of Hygiene and Tropical Medicine. But what's a common theme for all of us when we discuss Ita is how long our friendships and her mentorship lasted. I left her lab in 1984, and then had almost 30 years of her being a family friend and a critical and supportive mentor. So I think for all of those reasons, I will always be grateful to her. So the science today is really a much more recent event. This is work done by Lara Conejero and Javier Salguero in our lab and at the VLA. You can tell by what he's wearing is that I may be talking about a particularly nasty bug, but we'll get to that in a minute. And by Natasha Spink. It's a reflection of many years of collaboration with our colleagues in the molecular biology of the organism at DSTL Porton Down and in Thailand, where we've been working with for about 15 years with our collaborators there. And as you'll see later, more recently on microarray data with Anne and with Damien. So I'm going to talk about a bug that most people can't even pronounce, let alone know what it does. So I'm going to try and give you 15 or 20 minutes on what I think is an interesting organism. I work in a school of public health in the tropics. We don't just work on the big three, malaria, TB and HIV, and we're encouraged to work on things that are public health issues in the tropical environment, and this certainly is. So Burkholderia pseudomallei is the organism that we study. It's the causative agent of melioidosis, the disease in humans. You may very well know this genus of bacteria more from its related organism B. cepacia, which is a problem in cystic fibrosis patients and difficult to treat and manage. But it's more closely related to this really nasty bacteria, causative agent of glanders, which fortunately is very rare in a public health scenario. But if you travel into the tropical world, and in particular into the hotspots of Thailand and northern Australia, then this organism, Burkholderia pseudomallei, is a particular problem. So it was discovered by this gentleman here and his colleagues in Rangoon in 1911. He had the job of doing autopsies on the morphine addicts that had died on the streets of Rangoon in those days, and realised that they were dying not from a disease that he had already known about, but from a disease which was subtly but distinctly different. And he called it pseudomallei. The reality is that this is a fast-growing gram-negative bacteria. It lives in the soil and water. So if you're a tourist in Thailand, you could take an agar plate with you and isolate this organism while you're on vacation if you wish. But I would suggest that's probably not the smartest thing you've ever done. And what we find is that after all this time, we have genome sequences of the organism and beautiful cell biology done by others, which actually shows quite a lot of similarities to Listeria monocytogenes. It goes into macrophages, escapes into the cytoplasm, makes these beautiful actin comets, and here is the organism now searching to another macrophage in order to infect it. So there are some similarities in cell biology to Listeria, which is a paradigm that everybody's aware about, but as I'll show you, then there are many other differences. We work on this organism because it kills people in northeast Thailand. This is a hotspot of infection, high rates of sepsis in the rainy season from this organism. It takes almost as long to treat this organism as tuberculosis. If you survive, you frequently relapse even with the best treatments, and we currently have no vaccines that are available. As an immunologist, I think this is the slide that I hope tells you why we bother to study this organism, because what it describes is the huge range of clinical features that this organism can present with. And Western interest in this organism really came during the Vietnam conflict, when American helicopter pilots showed high rates of infection and acute sepsis with this bacteria, probably because they were getting aerosolized bacteria coming up into the rotor wash as they landed their helicopters in this area. But really, this is a problem of the rural Thai community. And poor people who have to go into the rice paddy during the seasonal times of the rice harvesting and planting. And these are the people who get exposed as children and start to seroconvert. So they get antibody signs of exposure in 80% of all the school-aged children in these regions. Now, most of these children will never get sick. But when they start to become older, in the ages of 45, 50, 55, if they have predisposing diseases, particularly diabetes, they run the risk of getting this acute fulminant cause of sepsis with a 40 to 50% mortality rate, even with the best antibiotic treatment that can be given right now. But interestingly, immunologically, in between these two exposures, there are people who have harboured this organism for 60 years or more, who have left the endemic area, aren't exposed again, so a fast-acting gram-negative bacteria can either kill you very quickly or hide away in your tissues for almost your entire life. To us, that's the immunological interest of this disease. So I want to talk about what it takes to generate protection against this acute fulminant pneumonia and sepsis, and I'll really focus on what is a rather strange observation, which is this organism is frequently misdiagnosed as tuberculosis in some parts of the world. So we don't get too many cases of tourists coming back with melioidosis in London, fortunately, and if they do come back, they tend to have cutaneous disease and not fulminant sepsis, which is a great bonus. So we have to work on mice. And we have mouse models of inoculation of the organism. The natural route, perhaps, because we think that most people in the Thai rice farming community get exposed by not wearing shoes as they walk around in the rice paddy and get subcutaneous inoculations. And then, of course, there's a biodefense issue here, where we believe that it's very easy for people to be exposed to this organism via an inhalational route. We have the usual genetics in the mouse of a genetically resistant strain and a susceptible strain. We have no idea what the genetics that underlies that resistance is. But if we put this organism into the nose of a mouse and generate a pulmonary infection and find out what goes beyond there, then we can model the acute septic events, giving 1,000 bacteria where all the mice will die within a few days of overwhelming infection. Or if we back off the infection dose, then we can start to get this very characteristic delayed course of infection, which actually starts to get around the same time as our tuberculosis studies. And I'm going to focus on a bit about that towards the second part of my talk. So what does it take to survive this first phase of infection and not to die immediately from this very fast-growing organism? Well, we think that there are two important innate immune events in mice, and we believe there is some evidence for that also in humans. So the first thing that got us into this was the fact that this is an exquisitely sensitive system to the production of interferon gamma. So these are mice that are infected IP or intranasally. The control mice will show this typical persistent phase of infection with a typical time to death of maybe 40 or 50 days. Gamma interferon knockout mice die in 40 hours. So it immediately tells you that there's a rapid innate interferon-dependent response. We believe it's driven mostly through natural killer cells, and it's driven mostly through the production of IL-12, because again, deletion of those molecules also causes acute and overwhelming sepsis. We also believe there's a neutrophil component in this early phase of protection, because when we do the best we can in terms of selectively depleting neutrophils with a monoclonal antibody called 1A8, again, you can see that these mice die rapidly compared to the control mice. This happens to be a lower dose of infection where the controls haven't died yet. We always work with our Thai colleagues who are studying these people in the endemic regions of northeastern Thailand, and they have found both NK cells and CD4 T cells that make interferon gamma in response to this organism, and the pathways of activation seem to be quite similar. And perhaps even more interesting, the diabetic Thais are the ones that are most susceptible to this disease, and they have clear defects in their neutrophil function and in their monocyte function. And those defects correlate with the inability to kill the bacteria and to migrate to the sites of the infection. So we think that there's a lot of parallels between the mouse and the human system. But I want to go back to this really annoying process by which this organism takes multiple steps in the time it takes to cause lethal infection. This is a mouse immunologist's nightmare, because it means that you have to use large numbers of animals. And it means at any particular time you have groups of animals which are either undergoing a severe event or are perfectly healthy. And the first step, of course, is to try to convince you that it's not because we're idiots and can't infect the mice appropriately. This is what happens when we inoculate mice intranasally with 100 bacteria. The mice synchronously increase their bacterial burden within 24 hours, and they're all essentially the same, as close as we can make them. But after that time, the mice then go off on this asynchronous path. Some mice will survive for long periods of time, and others will start to succumb gradually over the time course of the infection. So we also do mouse models of tuberculosis in the lab. This is clearly a strange event for a fast-growing gram-negative organism, and we wanted to try and understand what was going on in this more prolonged phase. And we thought we could learn a lot from tuberculosis in this system because, as I mentioned before, in parts of India and also I believe in Thailand, that this disease can be mimicking TB on chest X-ray. And you only find this out when you think someone has tuberculosis, you put them on anti-TB drugs and they don't get better, and then eventually you actually find out that you isolate Burkholderia from them. Also, Burkholderia has one of the key molecules involved in the persistence of tuberculosis, isocitrate lyase. And there's also this problem that the organism is persistent. The generation of sterilizing immunity is very difficult. And so treatment of human melioidosis, if you don't die within the first few days in the intensive care ward, will take almost as long as a classic 6-month TB treatment. So when you look at what goes on in our mouse models, we've decided to make a mouse model which was not the acute fulminant sepsis one, but was the chronic persistent, perhaps TB-like model. And so we gave low-dose infections into genetically resistant mice, and we tried to compare that pathology versus the classically reported pathology of acute infection in mice. In the acute infection, you see just death and destruction, huge numbers of neutrophils, very dominant pneumonia, and eventually sepsis, bacteremia, and death. But now, when we try to mimic this more chronic form of the human disease, the pathology is completely different. We see a macrophage-dominant process. You could see large numbers of multinucleated giant cells, which are never found in the fulminant disease. And we now start to see organised lymphoid aggregates. In the lung, we see very organised lymphocyte areas. We see, again, multinucleated giant cells, and then we get these lesions, which just look just like a TB lesion, with a central necrotic area surrounded by a cuff of macrophages and clear lymphoid aggregates. This is an area where you are failing to control the organism. There are high numbers of bacteria in that lesion, and this we see as a sign of failure of the immune response to control the organism. In contrast, when we look for bacteria here, we see very few. So this is a productive and protective response under these conditions. So what's going on in these lesions? And I'm meant to be talking in a macrophage-oriented session. Well, we are fixated on macrophage biology with this disease, since the organism preferentially goes into macrophages to live for long periods of time. But what's interesting is if you look at these lesions, they become very organised over the weeks in which they develop. And here is the central necrotic area surrounded by a very organised collagen capsule. So the host is trying to wall off this organism, we believe, and prevent it from disseminating out into the bloodstream and the tissues. And we see this organised granulomatous effect not just in the lung and in the spleen and also in the liver, which are the 3 target organs of the infection. We also think that there's a lot of macrophage activation going on, at least from Simon's talk discussing interferon gamma activated macrophages, classically activated macrophages. You can see massive numbers of INOS positive macrophages within the lesion. And they're quite interesting because they're sitting inside the collagen capsule and they're interfacing with the large numbers of bacteria which are sitting in that necrotic mass, which is in the centre of this process. So there's no doubt that there's a lot of macrophage activation going on. The problem is they can't kill the bacteria. So when we stain for bacterial components and not host components, and this is a stain kindly given to us by our colleagues at Porton Down, here is the control staining. What you see is a lesion, and the brown area is bacterial antigen. This is a polysaccharide capsule antigen from the organism. And so this site, although it's highly organised, has lots of macrophage activation and compartmentalisation by the fibrosis, is a site of massive bacterial replication. And you can see here is the central necrotic area, here are the macrophages loaded full of this bacterial antigen. And we often see some macrophages coming out loaded with the antigen, perhaps as they leave, I don't know. But here is the problem, and here is, I think, again, the interest. This is a typical set of 5 mice from a single experiment in this chronic, persistent, and heterogeneous disease. These are the key organs where we find the bacteria: blood, lung, and spleen. Those 2 mice have what we consider latent infection. We can't find the organism anywhere in those lesions. Yet we know it's there because if we wait long enough, the mice will all die. And they will all die because of an overwhelming infection in the spleen. So we just can't see it at that point, but there's somewhere in there. We also have experiments where we have mice which have, this is 2 by 10 to the 3, 6, 8, 2 by 10 to the 8 bacteria sitting in a lesion in the spleen, but there's nothing in the blood and there's nothing in the lung. So there's an amazing degree of compartmentalization of the organism at this point, but what we think then happens is eventually this breaks, this now leaks out, and the animal dies from overwhelming disseminated infection. And here is an animal where you see large numbers of bacteria spread through the different tissues and into the bloodstream. So to look at this process more carefully, in a different way, instead of keeping the beautiful 3-dimensional architecture of the lesion intact, we grind it all up and make RNA out of it and see what genes are expressed. And this is a very productive collaboration with Anna Gara and her colleagues and Damien Chasuble. And if we look in the lung early on in infection, in the acute phase of the disease, when the disease is very homogeneous and very tightly controlled, then we only need 3 mice to clearly separate the uninfected controls versus the mice which are infected with Burkholderia. And you can see there are large numbers of genes which are increased in their expression or changed in their expression during this disease. And we see a huge preponderance of chemokines, reflecting the fact that the lung is now trying to prepare itself to protect itself, and there's large numbers of neutrophils and macrophages or monocytes coming into the lesion. But what happens when we now do the same studies on the chronic phase is that now we get this very broad heterogeneous pattern. So if you look at a principal component analysis, which is basically separating the gene expression or the mRNA expression levels into its multiple components, then the uninfected controls all link together in here. The acutely infected mice all link together here. But the chronically infected mice show either a distinct pattern of mRNA expression, or they cluster right back in here where the uninfected controls are. And when we do the analysis of the tissue burdens in these mice, then we end up with mice which are latent, where we can't find any bacteria, and you'll see later that that's where these are. We have mice which are just emerging and are starting to spread out and become apparent to us. And then we have mice which are in these very severe and heavily infected bacterial burdens. And when we do this on a larger scale with a much larger number of animals, and we do again an analysis which is unsupervised, so we don't tell the computer what sample this came from, we simply look at the mRNA expression and rank it according to its closeness to each other. The RNA expression profiles are unique here to the infected animals. We cluster those very easily. In blue, you see the animals which are uninfected, and now you start to see these mice which were infected but now appear right in the uninfected controls. And these are the latently infected mice. So when we quantitate that by a weighted molecular distance to health method, you can see that compared to the uninfected controls, as the severity of the disease increases, we see this big change in bacterial burden and in RNA expression. But in the latent stages, we're essentially blind to this infection. We can't detect it by CFU, and we can't detect it by mRNA changes in the blood or in the tissues. We do have, from several years ago, a set of data where we collaborated with our Thai colleagues to take the peripheral blood transcriptional profiles of people in the intensive care wards in the hospitals in Ubon, Ratchathani, where they were looking at causes of sepsis, which included patients with melioidosis and sepsis with other organisms. And so we have peripheral blood microRNA profiles from the uninfected controls and from individuals who have either Burkholderia in purple or other bacterial pathogens. And we're in the process now of trying to take our different mouse models, both acute and chronic, and ask how good our mouse models are at mimicking the transcriptional profile of real people in Thailand who have got this disease and are presenting to the physician. That process is a prolonged one. It's involving a lot of people from Ganjana Alert Mamonkonchai's lab in Khon Kaen University, particularly Prasong Kenam, and people from Anagara's lab, in particular Christoph Potempa and Christine Graham. So in summary, this is a fast-growing gram-negative organism, and so initial innate immune responses are critical to protect against it, and we believe that's dominated by activation of macrophages by interferon gamma and by the recruitment of neutrophils. But this organism, unlike Listeria, which is this sort of beautiful paradigm of sterilizing immunity, this organism just doesn't generate a sterilizing immune response. People harbour the organism for their entire life, and mice will eventually succumb without being able to clear it. We think our model that we've generated now mimics the pathology of human melioidosis more closely, and it actually shares many of the features with tuberculosis. We see areas of central necrosis, we see giant sheets of epithelioid cells, multinucleated giant cells, and even some foamy macrophages, all from an organism which, in other circumstances, can kill you within 2 or 3 days. Finally, our transcriptional array analysis shows distinct immune gene expression between the acute and the chronic phases, and, as far as we can tell, silence during the latent phase. And that is an area where we would like to focus more of our attention. So to finish off, what do we know or not know? Well, most of it we don't know. This lesion, we believe, is really the central point at which we have to control this disease. It's the point where we are failing to generate sterilizing immunity, and it's the point where we are unable to eradicate the bacteria by antibiotic treatment. We would love to know how the fibrosis that I showed you that encapsulates this lesion is generated, and whether it really does help reduce the dissemination of the disease. We would really like to know why the macrophages that are holding all of these bacterial antigens just cannot kill the organism, yet show many signs of activation as far as we can tell. And what we'd really like to know about is how do the bacteria feel in these lesions? Because the metabolic state of the bacteria in those lesions will probably greatly affect its susceptibility to killing by both immune responses and by antibiotics. And as I mentioned before, we're about to do an interesting study, just trying to see really how good our mouse models are in comparing them to the human infections in northeast Thailand by comparing their blood transcriptome analysis. So I think I'm on time. Again, thank you for the chance to speak here and to remember Ita, and I'd be happy to take any of your questions.
Questions for Greg?
Is there a natural host for this organism, just a soil organism, or does it live in other living things?
Its natural host is probably a rice plant, but you can grow it in amoebae. So it may well have learnt a lot of its tricks on how to be inside macrophages and phagocytic cells by having to coexist with soil amoebae.
And in the rice plant, does it kill the rice?
No. Doesn't have any pathology whatsoever.
So what's rice got?
We are unlucky enough to be exposed to this disease, this organism, and we are the end stage. Because unlike tuberculosis, we don't cough it on to the next person. So,
Do they die in the rice plant?
do the bacteria die? No. The bacteria coexist with in the soil with the rice plant.
Is it worth looking to see if the rice is producing something positive in shape?
There's a lot of people interested in phages, because there are lots of phages in the soil in this area, and you can find different parts of Thailand which do or do not have the organism. So there's a lot of people interested in looking at the environmental ecology. But I'm a mouse immunologist, so that's not the area that I'm following. So.
Liz?
Can I ask a question as a mouse immunologist?
Oh, yeah.
Can you transfer the latent state or the propensity to develop a latent state by taking any population of lymphocytes into mice that have got very low levels or are given very low levels of the bacteria?
Now, we've never done any transfer experiments at all, and I guess you'd like to purify macrophage populations and various cell types from those animals and move them across.
It's not even impossible that there are other cell types that are acting in a regulatory fashion.
Yes, it's entirely possible. I mean, this organism will go into other cell types. It's just that it's the macrophage where we tend to find it most dominantly.
That doesn't, the regulatory process doesn't necessarily need
No.
to be in the macrophage.
No. I'm expressing our ignorance of knowing where it's hiding and what actually causes it to reactivate, other than the fact that diabetes is a clear risk factor for the organism.
Andrew.
So there's misdiagnosed in humans diagnosis for TB, but you ended up giving 4 antibiotics for TB, or at least 2 humans, and then 2 antibiotics for 6 to 12 months. Is this high resistance?
It just tends to be highly resistant to every other antibiotic, so you have to use a select set, which is imipenem and ceftazidime, intravenously for 2 weeks. So it's not generically, fortunately, you can still treat it this way, and there's no resistance to those antibiotics. It just takes a long time to eradicate the organism. I mean, just as it takes for TB, it takes only a few weeks to clear many of the bacteria from your sputum, it takes a long time to get rid of those last few persistent organisms. It's a really good question. I mean, a lot of people will relapse under these antibiotic treatment conditions. So under those situations, they've gone through their treatment, they've appeared to get well again, and within the next 12 or 24 months, they come back again with Burkholderia. Much of that is reactivation. So what that really means is you haven't got rid, I think, of the very last few organisms. To me, that tells you that you've gone through this process. You are not immune now from having made this big immune response during the active infection. So I don't think pre-existing infection protects you very well against the disease.
Dave.
But in the mouse, are neutrophils a persisting feature of the granulomas?
In the acute phase they are, but not in the more chronic phase where we start to see a much more macrophage and T-cell oriented phenotype. So it just depends on the kind of intensity of the infection and the genetics of the animal you're looking at. And in humans, we see both a combination of neutrophils and granulomatous macrophages. So it's a pyogranuloma in a sense.
The last question before lunch, Nick Wilcox.
Whether antibodies would be beneficial for the bacteria. We can passively transfer protection by antibodies. So I think it's probably not a good thing to make from the bacteria's point of view, not a good thing to be making antibodies, no.
Okay, Greg, thank you very much. And could I implore you to come back by 1:30 for the start of the next session? Thank you.